infect the plants or the disease or the spread of a disease. Number one, we just carefully inspect seeds, bulbs, plants before planting, especially case of a root system. And also in our pathology, in your field condition, you should remove the plant debris or senescency, because our pathogen, for example, rust spores rust are present when the field condition wheat stage is a most appropriate condition where the rust will be able to attack. When the wheat is not present in our crop, in our field, where does it goes? Kahan jayenge? So it should be harbors on the debris. It should be harbors in the weeds. It should be harbored on the standing crop, which should be serve as a parasite. So it should be very clear that we should remove the plant debris after harvesting the crops, and also the remove or prune the infected plants. And we all you can say scheduled walks to the garden on a regular basis during the growing season. And the most important thing, during the sanitation or IDM, use clean equipment such as pruning shears, flat pots and space, because most of the pathogen can transmit from one place to another place by using by used equipment such as nematodes, such as mostly fungi spores, conidia, sclerotiums. These can be spread from one place to the other one. So, and the important thing for all this assessing the disease, for diagnosis of the diseases and the management for the diseases, is the record keeping. What will happen in the next year? For example, I just my team was shown the food form maize, and we sown the pulses. In our last year, before last year, we saw that we observed that the many diseases are occurred during this year, but not the last year. So record keeping is important. And I also observed over there that the one land that where the wheat was sown, but one land is there where the wheats were more abundant in amount rather than the adjacent area. So I just find out that this means that in the adjacent area there is some kind of the debris over there, or their seeds should be there. So this is the continuous research and continuous record keeping employed for the sanitation practices also. And we also, in an IDM approach, we also use the use of resistant cultivars and resistant varieties is very play an important role for them. If you say the most important, most prevalent pathogen or a disease in vegetables in a Potohar region is the powdery mildew, recipes. Powdery recipes prevail them. Cucurbitaceae family, like watermelon, like cucumber, like aap kaddu keh le, ispe pumpkins hai. These are the most growing vegetables in our Potohar region. So if the resistant powdery mildew, if the powdery mildew, we just see that this variety shows this much amount of the incidence of inoculum over there. So we should replace these susceptible varieties by the resistant variety, such as you can say that if we in a Cucurbitaceae family, cucurbits mostly, against the powdery mildew AS454, Hijazi, and CU19 and CU15, and if you most prevalent, you can say most growing crop in our in the field in the Potohar region, wheat is there, and the Fakhri Bhakkar, Chakwal, and Zincol. This is also a resistant one. And if you say about the Verticillium epizerium wilts, which is a very most important in this area wilting, because two types of wilting over there. One is a temporary wilting, and another one is a permanent wilting. Temporary wilting is due to the deficiency of a water, and we can just recover it by giving the water to our plants. And if this permanent wilting is due to some kind of a bacterial infections, and vascular bundle will be showed by the presence of the bacteria. So These can be recuperable by temporary wilting, may be recuperable by the giving the water to them, or Fusarium is like Fusarium is a soil-borne pathogen, Verticillium is a soil-borne pathogen, and Phytophthora is a soil-borne pathogen. These pathogens are a soil-borne and can be, you can say, by the can be using most kind of a pesticide, fungicide. It can be removed, but the resistant varieties may a major role for it. And bitter boy and celebrity, these are using for this. And the groundnut is also a most important crop of in our Potoha regions, and Bard 479 is also we are using for the for it. Now let's come with a important point for the eco-friendly management approaches. We use of different living organisms and their byproduct for the control of plant diseases caused by the other pathogen. Like we are, this is you can say it is a more safest, non-toxic, no residual effects, no environment, no effect on the environment, and pathogen can control easily and naturally. If we compare this biological control to the chemical control, it is very toxic to nature, residual effect on human and animals, affecting the natural resources, soil health को भी खराब कर सकता है, and this chemical control may produce a resistance in the pathogen. So if we say that how this mechanism of the biological control agent play, that is first of all is a parasitism. Parasitism is the organism feeds on the pathogen, right? We can say that it is a parasitism, and it may be antagonistic, may be due to parasitism. And microorganism activity in this manner are known as a hyperparasite. So the microparasites may be necrotrophic or biotrophic in nature. The necrotrophic may or may not infect the host but kill it by producing a toxic substance or salicylic enzymes and nutrients that release from the dead hyphal cells, which are utilized by the mycoparasite. On the other hand, we are comparing the parasitism between the microtroph and the necrotroph. On the other hand, the biotroph by establishing the intimate contact with host cell or by penetrating them and obtain the nutrient without killing the host. So this is the parasitism. The ability of the pathogen to symbionts to live with the other organism. उसमें उसका फायदा भी होगा, उसमें क्या होगा कि उसको एक का नुकसान हो जाएगा और एक का क्या हो जाएगा, एक उसको इंटेक कर लेगा. Now the second one is a antibiosis. That is important. Induced resistance is also important. The organism activate the defense mechanism in the plant. We just accelerate the salicylic acid and we just accelerate the defense mechanism of the plants. Now, what about the antibiosis? Antibiosis, the organism secrete toxins that inhibit the pathogen. How it's work? Like microbes may produce different kinds of antibiotics capable of inhibiting the growth of the pathogen. We can take an example. Gladiolinium varians produces a powerful antibiotic, gliotoxins and viridin. So this is the antibiosis. क्या चीज है उसमें, ऐसी क्या चीज कर रहा है? That is that secreting the toxins that inhibit the pathogen. What about the competition? Because we are just talking about this, the biological control. So we will be moved towards biological control, towards the biological control agent. So briefly here, I just want to describe what are the mechanisms behind this. One is a parasitism, the other one is induced resistance, the third one is antagonism, and the third one is a competition. How the competition between the two organisms will be prevail. So this is the competition. One pathogen might be inhibit the second one. So the competition just prevails. So for the work of the biological control, we have a clear picture about the mechanism of the biological control and the biological control agent. So in the biological control agents, in a biocontrol, because we are working on our plant pathogens, and we where is the these pathogens also work as a foe as well as a friend fungi, friend one. So first of all, we take an example of a fungi. Most fungi behave like a friendly towards the other organism, also viruses, nematode, and bacteria, and also plant extracts and other essential oils. They also working as a biocontrol agents, and which will be used for the controlling of our disease in pre-harvesting, after harvesting, post-harvest fruits. So these can be used as a biocontrol agent. Let us talk about the trichoderma. Trichoderma is a fungi, and it is also used as a biocontrol agent. Biocontrol agent that is present in all soil. Trichoderma, like you can see the pictures, it's a conidia, and it's attachment of a conidiophore, because we cannot identify the fungi without the attachment of the conidia on the conidiophores and the other parameter. And if you can see the above picture of them, is a cultural cultural appearance of the trichoderma in our petri plate, and in the next you can say B, C pics, B, C, and A, B, C, D pic, we can say the conidia, and the attachment of the conidia on the conidiophore we can easily see over here. So then if you just trichoderma is used as a biocontrol agent that is present in all soil. It produces many antifungal metabolites, including antibiotics and cell wall degrading enzyme. Like we every pathogen, every plant, yeah, you can say that the plant is growing and nourishing and giving the fruits. There is a physiological functions that the plant is taking, and all the things are happening. So if the pathogen we are controlling the pathogen for the disease, so what will be happening behind them? This means there is some kind of the mechanism that the pathogen control this kind of the fungi is, yeah, you can say biocontrol agent is control the other things. And what is the what is the things behind them? How can say that? You can say that if trichoderma is a fungi, that there is some kind of antibiotic, trichodil, and the cell wall degrading enzyme that is involved in it, and that suppress the disease, that suppress the pathogen, and minimize the disease losses and the incidence. So this is a little bit information about them. I would like to share the work which my team and my students I'm going to be doing in my lab on the essential oil. You can say I just give all the information and all the integrated approaches and friendly approaches, IDM and chemical control and the sanitation and all the things. But basically, our target is to control the diseases, plant diseases, for the very safest, very cheaper, and very easy to handle and very easily available for our farmer. And you can say that our research, our experience should help our farmers, our orchards, our poor people, that they can be able to improve and they can minimize their losses, they improve their product, their yield. So there is an entire need of for this era is a biological control, and I think all over the world is using this type of a product, but we are just lacking them. We are just behind of them, and we should also work for them. And so we select this essential oil. So essential oil for the management of the plant pathogens. So what is the essential oil? And essential oil is concentrated hydrophobic liquid containing volatile chemical compounds, also known as the volatile oils, ethereal oils, simply as the oil of a plant from which they are extracted, such as ऑयल ऑफ क्लोव। लौंग हम अपने घर में कितना अच्छा यूज करते हैं, अपने दांत के दर्द के लिए यूज कर रहे होते हैं हम। उसको जो है वो उसकी जो है वो उसमें अपने पुलाव बनाने में यूज कर रहे होते हैं। अगर आप दालचीनी को देखें, वी आर यूजिंग अ सिनेमन एंड वी यूजिंग अ इलायची, बड़ी इलायची, और हम इसमें जीरा, लाइक so many spices and so many condiments, and we are using in our all the you can say in our kitchen, or you can see easily the seeds of the our theme, our basic idea was that the useless things like the seeds of the citrus, peel of the citrus, and you can say seeds of sumbul seeds, like the flower of the gladulus, like these are tulips, so you can say not a flower, it's a tulip. So these things can be used for the management of the diseases and controlling the you can say minimizing the loss and increase the production. So our basic theme was that, and we used and we targeted for the essential oil. So essential oil are produced and secreted by granular proteins, specialized secretory tissues, and diffuse onto the surface of a plant organ, particularly flowers and the leaf. So we use not these, but this is only the small list of the plants we use for this. My students, nearly more than 15 students and 3 PhD students work on it, and one of them use cannabis, bhang, carom, fennel, thyme, brassica, eucalyptus, ginger, garlic. These, and the most important thing for is that the all part of the plants are not be able to secrete or having a essential oil for them, such as the cannabis leaves, cardamom seeds, fennel seeds, thyme leaves, brassica seeds, eucalyptus leaves, ginger rhizomes, and garlic clove. So we use these seeds for the management of the plant diseases, especially post-harvest fruits. Now, essential oil, as our history is that it would be a essence, and it would be used in a perfume industry, and is used as a essence in our cooking foods. So it will be very old history as in a Rome and a Greece, but in our subcontinent it will be started in 1923, and then in the next one is in 1983. Monoterpies and phenolpropanoids have been reported, and that would be an important constituent of a resume essential oil, that would be a clove. In 1997, several different species are distributed and identified all over the world. But the important thing, the use of synthetic insecticide which pose health hazards and are largely incompatible with subsistence agriculture, reported biocontrol against the diseases. So this was the new era that these essential oils can be used as a fungicide biocontrol agent, and would be able to the next for the new researcher, new students, for the students also. And so it will be a result of new compounds, and would be and essential oils have been proved to be inhibiting against a wide range of food spoilage microbes, dependent upon the concentration, method of testing, and active constituent. Now, what are the antifungal activity? Because we are interested in our like this. We are facing the post-harvest losses in fruits in Pakistan, and nearly 35 to 40% of our post-harvest losses have been reported in the Pakistan, and most of the cases these losses are due to the fungi. You can see easily that citrus. I have also different pictures of the grapes and locust and peaches and apricot, but the most prevalent where the mold can see easily, that is on the citrus. Like green mold, you can see easily that it's a velvety growth and velvety and greenish whitish growth over the fruit, and it's a bluish whitish on a growth, and it is a green mold, blue mold, gray mold. Green mold caused by Penicillium, while blue mold caused by the Penicillium expansum, gray mold caused by the Botrytis cinerea rot. You can say we are also facing some kind of rots on the grapes, and most of the bunches will be a very, you can say, all the demander will not be able to just take, हम उससे hesitate ही करते हैं कि we cannot be able to purchase them. You can see is a black rod, and in a peaches you can see it's a rhizopus rod, and anthracnose, and a black rod and sour rod. One of the causes that the green mold, blue mold, gray mold, that the fungi are involved, and the other when we just eat this time we eat food, most of the Aspergillus niger causes the aflatoxin B1, G1. This is also a very insidious to the human health, and most of the cancer, breast cancer, hepatitis may also just correlate from them. So one of the choosing the biocontrol agent is to control this, like to reduce the losses, and also the to save our human health. Increase the productivity, increase the yield, and improve our human health. So this is just for the information about the rotting, which we are having we are facing in our field conditions, in our market, and also in our houses. So the essential oil, the antimicrobial activity of essential oil is associated with the presence of secondary metabolites synthesized by the plants. And its mechanism would involve that the interaction of its hydrophobic components with the lipid present in the cell membrane of the microorganism, resulting in the metabolic damages, and they cause the cell death. So this is a little bit antifungal activity of the essential oil. When we perform this all screening of essential oil in our lab, so we just choose with all the essential oils not play an important role in having antifungal activity for the pathogen. For example, if we just perform a practical, we can say in our PhD research, thyme is very important. You can say it's play a significant role against the Botrytis cinerea, gray mold, causing a gray mold. But other phenol and cannabis not play an important role because they don't have an antifungal activity. So it's not mean that all the essential oil have the antifungal activity. One of the, you can say some of them have the antifungal activity, and which play an important role for the controlling the disease. Because we are just focusing our biocontrol, and that product will be able to in the market and help the farmers to be able to enhance their production and to minimize the losses. So that's why we screen all the essential oil in our lab against the pathogen, and they just show if for this how can we able, like if there is a green mold, blue mold, gray mold, alternate mold, for this first step is the isolation of the pathogen. If we say that this is a green, like it is a it's a condition of the citrus fruit, you can see that the velvety growth, greenish, whitish growth are there. How can you see that there is some important keys, identificational keys, which is we are using a Listeria journal of fungi, and some important, I think the book name is Dimetaceous hypomycetes. We are having a different type of manuals over there in our lab, by which we can just identify, we isolate this pathogen. If you say that like this type of fruit is suffering the problem, we isolate. We just take a sample from the field and just and process under the different protocols. And first of all, if we just only take an example of a green mold, how we isolate them? We isolate them by adopting isolation techniques. We just we are having a morphological and a molecular characterization. हमने उसकी तरफ जाना है for the management practices के लिए. उससे पहले हमें ये पता होना चाहिए that it is a It is a Penicillium expansum, and we are sure about that it is the Penicillium expansum या digitatum. So for this, we have to we have to adopt all the techniques. How can we perform that? If we are infected fruit, infected plant, so what will you do? That you just take a sample and just place in our lab, in our fungal plant pathology lab. अगर bacteria का sample है तो you will proceed for a bacterial lab. nematode का है तो जो fruits और में ज्यादातर जो problems है वो bacterial और most of the cases जो है वो ज्यादातर fungi के. So you just your fruit just take a sample, and before sampling you just have to, all the things should be ready in your previous condition. Like if you just go to the take the sampling in this morning, so your all the things like petri plates, like media, like all the things should be ready in your laminar flow, yeah, in your fridge before sampling, yeah, before starting the work. You just take a sample in your lab. Now, what will be happen? That in our laminar flow, you just sweep, you just all the should be clean with your spirit, and just on the UV light and just on the light, and then you have to just out of the you just go away from them, and then you just take a pits of the section from the margin of a lesion, place in a 10% of a Clorox for different duration, and it is a protocol, and for the plant pathologies it's a very simple that all the process have been passed on, and sterile forceps used to transfer sections, and these will be a tissue sections dotted with a sterile paper towel to remove the Clorox excess, and sections are placed on a nutrient media in a petri place. Now, after the 7 days or 10, you can say the incubation is a very important time, because it is a time period between the entrance of the pathogen and the periods between the and the period of the pathogen. So we just gave the conditions and a suitable temperature and the nutritional requirement for the pathogen, and to for their colony, for their identification, morphological identification. And if you see that the same as the process in a bacteria, that in the fungi isolation we use a PDA, and in our nutrient bacterial identification we use a PDA for this. So these are the important features for the identification of the fungi. Suppose culture color. You can see the first pic in the petri plates is a culture color, spore size, fruiting bodies, and we compare these keys. This is the reference given below over here. Ellis and Lauren, 1998. We use though. It is an important identification keys for the morphological characterization of the pathogen. Whichever, either we just isolate from the food, either we just isolate from the leaf, either we just take from the roots. Either this is either a foliar pathogen, root pathogen. This is an important component due to the basic component from which we can say that it is a we can identify, and for the diagnosis it is a important parameter. You can say it's a basic parameter for this. Now come on a point that identification of the pathogen, identification keys are there, and morphological characterization. And one thing is missing over here, that I want to share that these conidia are of course just like I know that it is for alternaria. More species alternaria are there. So what would be, we can say that how can we identify that it is a alternaria, alternaria, capsicum, alternaria, capsii? How can we identify? We use ocular micrometry, by which we can be able to differentiate from one species to the other species. So as we are going to be discussed over here as a biological control agent, and one of the control agent we are using is essential oil. How can we extract in our lab condition? So I think in our 2016 or 17 I purchased this oxalate from one of my small project funded by University of Agriculture, and I install it, and this is a oxalate extraction apparatus which in from which yeah, you can say it's a principally work on evaporation, condensation, and extraction of the. We are focused on the essential oil. Like if you are just working on a cannabis, if you are working on a bark of a eucalyptus, this is depend upon the what will the targeted essential oil are, what will be the targeted our host. For this, we take a sample and let them dry in our room temperature, and might be some seeds of the plants may be dry one day, might be it will take a 2 days, it will be take a 3 days, so it will take a time. So dried seeds to dried peels, like one of example is a citrus peel, and it will be dry more than 4 years, 5 years, and we should have to dry in our also sun dry. So we dry them, and then we use ethanol and methanol or for the solvent. In the you can see that it is the lowest part is a there is in which a round bottle or face is a mantle, we say is its whole part is called as a mantle, and that round bottle in which we place our solvent, and you can see it's the above portion is the condenser in which we place our dry sample over there. What will be the principle behind this? When it will be attached with the distillery, and from one place the water is just going on inside of the round bottle, and in which the solvent is present, and evaporation does play important role. Vapors will be condensed and comes with the essential oils with them, and again it will be collected in a round bottle, and it will take 6 to 7 hours. It depends upon the condition. It depends upon our host, from part of the host from where we are just taking the our essential oil. If we are working citrus peel, so it will take 2 to 3 hours. If we're working on we want to take essential oil from the seeds, it will from the citrus, it will, I think, more than 5 hours will be take. So it depends upon the host. Aapka host kaun sa, then it will be able. Now, first, essential oil, selection of the part of the essential oil, like if you're taking a plant, a seed, a plant mein kaun sa plant hai. So these are the selection, and how you manage the disease. I also mentioned that all the essential oils of the food, all the essential oil will not having antifungal activity. So after screening the essential oil, we will be able to the plant pathogen, will be able to use in a plant pathogen. We will be in literature, there are so many methods are available for the management of for the managing of the disease in vitro condition, but in our lab we are using food poison technique, and in poison food technique in vitro evaluation of plant essential oil against most prevail decaying causing fungal pathogen by preparing different concentration. It will depend कि आपने क्या चीज यूज करनी है, कैसे यूज करनी है. So it depend upon the condition कि कौन सा आपने मेथड यूज, कौन सा आपने होस्ट यूज किया और कौन सा आपने उसके अंदर वो यूज किया. The percentage of mycelial inhibition measures MI percentage measured 3 days after the incubation, and it will be calculated by using the following formula. DC, यानी the diameter of colony divided by DT multiplied by DC. This is a specific formula. One is that you just not treat with the fungus, you just not in poison food techniques we also use 2 methods. One is a swab method, because it's a volatile compound, so we use a Petri plate, and in which first in a media we just pour our essential oil in the media, and then we cover it. This is the one method of poison food technique, that we mix the essential oil in all our petri plates. This is also a technique, but in our lab condition, we also use another method, that we just dip the swab in essential oil, and then we place on the lid of the petri plate. This also play a very important role, but there is a lot of contamination I face over the period of time. So I just recommend that poison food technique is a much better as compared to the other one. This is now in it. DC diameter of colony. In the first petri plate, you see that the fungus colony would be determined by the scale, by the ocular, by this. In a conventional methods, we're using a scale. On the petri plate, we use a scale and we determine the diameter. Where from the DC and from the colony of the diameter of colony divided by diameter of treatment and diameter of the other colony. So by this we multiply by 100, by which we can be able to determine the how the which essential oil having a most prevalent antifungal activity against the pathogen. The other method we also use in our lab is a well diffusion techniques. Agar well diffusion is reliable technique used to screen out the antifungal from plant essential oil. 2 wells were made with a sterile cork borer and education from each other, and other well were filled by the pathogen. From the targeted pathogen, for example, you are just taking a Botrytis cinerea causing a bunch or grey mold in our grapes. So you check the pathogen and a limited amount of inoculum just fix in just pour in a one well, and on the other well you just take our PEOs, plant essential oil. So diffuse in other media which inhibit the growth pathogen. Then you will, how can you determine this type? How would be able to take our data? Like if the essential, the borer, the well, 2 wells over there in the petri plates, one are having a pathogen and other are having a essential oil. If the essential oil have inhibit the fungal growth, this means that the essential oil is having antifungal activity. If the essential oil don't have any inhibit the fungi, this means it has not any fungal activity. It depends upon the diameter of colony diameter on the petri plate, by which we can differentiate, we can screen out the essential oil by 2 techniques. One is and for this is a linear growth inhibition was measured after 7 days, and mycelial growth of pathogen was calculated by that formula DC divided by DT multiplied by DC multiplied by 100. This is the you can say it a common formula and can be applied for all the techniques. So by this method, first of all we discuss essential oil, then we discuss how we screen the essential oil for the management purposes. We use 2 methods. One is the well diffusion poison food technique, and the other one is a well diffusion method. For the you can say that we screen out the essential oil, all the essential oil having a like out of 10, 2 or 1 or 3 are having a antifungal activity, and remaining of them have not the antifungal activity. So either they have the antifungal activity. So what is the active ingredient for having an antifungal activity? So we perform, although it's we can say we use a HPLC gas chromatography for this, but for the cheaper, for the easier in our lab, we use a different phytochemical analysis test like antifungal compound, terpenoids, alkaloids, phenols, saponins. For the terpenoids, Silkowski's test is there. If the all the protocols followed by this test, and if the dark blue conditions are there, the dark blue are there, so we say that the terpenoids are there. If the alkaloids are there, the dark brownish to black color indication. So phenols are there. If the phenols are having occurrence of phenolic at their test is a test performed, we can say it's a ferric chloride and yellowish to green color. Saponins and saponin test, then a form like a penis would be there. So yeah, this is the very cheapest and easiest methods for checking the antifungal compounds. The last step, we like in our labs we perform is extraction, then we perform screening, Then we performed either the antifungal that they're having a antifungal, which compound is important and which compound is having as serve as an active ingredient. Now the question is that this application of the essential oils, how would we perform in our fruits in our in vivo condition? For this, the selected fruits are available, and we them wash, and healthy bunches of fruits were washed and sterilized with the water, and will be surface sterilized with a 0.1 sodium hypochlorite solution, and again washed with the distilled water. The fruits were inoculated by 1 ml of standard spore suspension of a major pathogen. Here is the one should be clear. There is a one should be clear in mind that the standard spore suspension. Every pathogen having the different spore suspension. Like for the Alternaria, but
Hello. Hello, Dr. Gulshan, can you please hear me? I think there might be some problem with the internet. Hello. Please let me check with Dr. Gulshan. Hello. I think there is a technical problem, and she will be again in touch with us in 2-3 minutes. I would request all the participants to please wait for a while, and she will be back to